Hi everyone, welcome to the third and final lecture over the second part of Chapter 21, the immune system. In this lecture, we're going to be wrapping up cellular immunity with cytotoxic T cells. We're going to talk about humoral immunity with B cells, and we're going to look into vaccinations, allergies, and some immune disorders. But let's start with our not really attendance questions. Why does someone with type A blood have anti-B antibodies? How do anti-B antibodies recognize type B blood as foreign? And what do NK cells do when something is recognized by them? So go ahead and try to answer those before listening to the correct answers. And let's see what those correct answers should be. Why does someone with type A blood have anti-B antibodies? Well, remember, antibodies, part of our immune system, are looking for something that does not belong in your body. So someone with type A blood has A antigens on their erythrocytes. They should never have B antigens in their body. So anti-B antibodies are on the lookout for B antigens because those B antigens would be out of place. They would be foreign. So how do anti-B antibodies recognize type B blood as foreign? Because the ends of those anti-B antibodies are shaped so that they would fit the B antigens that would be on B-type erythrocytes. And they would fit like two pieces of a puzzle. What do NK cells do when something is recognized by them? Well, first, how do NK cells recognize anything? Remember, NK cells, natural killer cells, are part of the innate immune system. They are non-specific. So they don't have receptors looking for something very specific. They have pattern recognition receptors that just look for patterns in antigens, something that looks like a bacteria, something that looks like it should not be in your body. And when something sets off those pattern recognition receptors, the NK cells recognize that as foreign, and it will start to release things like perforin and granzyme. So all three of these things are going to come up in today's lecture. And let's go ahead and get rolling with that. At the end of the previous lecture, we had gone over this slide, and we had started drawing some T cell activities. We drew what the CD4 helper T cells do, and now we're going to wrap up and draw what cytotoxic T cells do, the CD8 cells. So let's go ahead and switch over to our whiteboard. And this should look very familiar. Across the top, the CD8 cells, remember these are naive, they're mature, but they are still naive. And they really look a lot like those CD4 cells that we drew. They still have that receptor that's got that weird arm on the outside of it. But CD8 cells, the cytotoxic T cells, they are going to respond to cells that are either cancerous or virus infected, or that are transplants. And that's what we're going to draw here. Remember, the CD4 cells, they responded to antigen-presenting cells that are coming along and showing, hey, I just ate this, we should get everyone ready to fight. CD8 cells, though, these are going to respond to something a little bit differently. So let's go down here and look at what we've drawn down here. This is a body cell. And on the surface of this body cell, it's got an MHC. This is MHC class 1. Remember, MHC class 1 is on practically every cell in your body. And this cell that we've drawn, maybe it's a cancerous cell. And remember, once a cell becomes cancerous, it is cancerous forever. And when it divides, it produces 2 cancerous cells, which then divide to produce 4 cancerous cells. Or instead, maybe this is a virus-infected cell. And virus-infected cells, once they're virus-infected, they are virus-infected forever. There's no fixing it. And when it divides, it produces 2 virus-infected cells, which then divide to produce 4 virus-infected cells. Now, if we're talking transplants, it's going to be similar to what we draw here, but it's got some extra stuff that we really don't need to worry about. So we're just going to talk about what would happen in the case of cancer or virus infected. Now, if this was a cancerous cell, this cell is going through mitosis when we really don't need it to go through mitosis. If this was a virus-infected cell, it's busy making copies of viruses, but not doing so much of what it needs to be doing. Now let's go ahead and talk a little bit more generally just about what this cell is doing normally, and then we're going to come back and talk about virus and cancer. On the surface of every cell in your body, there are lots of proteins. We are specifically going to look at MHC class I proteins. Proteins on the surface of a cell don't remain there for very long. The outside of a cell is kind of an inhospitable place. So when a protein moves out onto the surface of the cell, it stays there for a little while, but then it's degraded, or it's destroyed, or it outlives its usefulness. But then this cell is constantly making new ones. So an MHC class I protein is made, it moves out onto the surface of the cell, it's there for a while, it goes away. And then this cell makes more MHC class Is, which then move out onto the surface of the cell to repeat the cycle. What that really means is, at any one moment, the MHC class I proteins on the surface of a cell are relatively new. Okay, now let's look at a cancerous cell. If a cancerous cell is going through mitosis pretty much endlessly, during mitosis, other things are kind of put on the back burner. Cells are not really doing all the stuff that they normally would be doing. So it still makes MHC class I, but it's not devoting all of its resources to it like it normally would. And so a cell that is cancerous will have MHC class I on its surface, but it's going to be shaped a little wonky. In the case of a virus, so let's say this is a virus-infected cell. Let's go even better. Remember when we drew interferons? And that one cell that we said was a lost cause, there was no helping it, it was going to be forever infected by a virus? Let's go back and say this cell is that virus-infected cell. Remember, he warned his neighbors, but there was nothing that we could do about him. So this cell is virus-infected. It's preoccupied with making more and more and more copies of that virus. So every time it makes new MHC class Is and moves them out onto its surface, well, this cell is filling up with virus particles. And when this cell makes an MHC class I that moves out onto its surface, it's going to have a little bit of that virus stuck to it. So really, an MHC class I doesn't just identify this as self, it also gives an idea of the state of this cell. By looking at the shape of this MHC class I, it says I am a body cell, and this is my current condition. In this case, this MHC class I says I am a body cell, and I've got something that looks like this inside of me. So now we know not only is this a body cell, but it's got something going wrong with it. Maybe it's virus infected because it's shaped wonky. Maybe it's virus infected because it's got a piece of virus. Maybe it's cancerous because it's shaped a little wonky. Whatever it is, this MHC class I has something going on with it. So This CD8 cell, bumping up against every surface in your body, comes along, does its job here, doesn't recognize anything, and moves on, still naive. This CD8 cell comes along, does its thing, checks it out, doesn't recognize anything, leaves, still naive. And that keeps going on. But at some point, this CD8 cell is going to come along. And when he does, it's almost like we drew yesterday, or last lecture, with our CD4 cell. We've got this arm that reaches around and says, hey, I see this is an MHC class 1. You're probably going to show me something. Let me see how things are going with you right now. This arm reaches in and says, oh, so you've got something wrong with you. Let me take care of that. And this cell is going to start to divide. And some of what it divides into are are memory cytotoxic T cells. They don't do anything right now. They go hide, they hang out in your lymph system waiting for something to come up later. And we produce effector cells, the ones that are going to be active during this fight. And these are called cytotoxic T cells. Now, even though we are drawing this as it happens, there is a catch. This step right here, when he's dividing and differentiating, that requires helper T cells. Really, after this recognition occurs, this cell, while it is active, but before it starts dividing, it goes and finds a helper T cell, shows the helper T cell this antigen, and then the helper T cell blasts him with interleukin-2. Interleukin-2, remember, is required by every immune part of your system. So this happens, but really it happens because of interleukin-2. So once we get these effector, these cytotoxic T cells, what do they do? How do they respond? Well, it's actually very, very similar to how the NK cells worked. They're going to release perforin. Perforin is going to perforate, poke holes in this cell. They're also going to release granzyme and something pretty similar, we're just going to call it the same thing, called lymphotoxin. They're not really the same thing for purposes of this class. We're just going to call them the same thing. Perforin pokes holes, granzyme leaks in. The granzyme, like we saw with NK cells, starts to trigger apoptosis. And that will ultimately kill this cell. Also, the TC cells are going to release something called interferon. We won't worry about which numbers. We're just going to say interferon. Basically starts immune cells to become active, just like we saw with helper T cells, but it does it a little bit differently. And it's going to release something called tumor necrosis factor. And tumor necrosis factor is anti-cancer. It's anti-carcinogenic. It's going to start to focus on tumor cells. So, remember helper T cells, they didn't really do any fighting. They just told the rest of your immune system to fight. But cytotoxic T cells, they do some killing. They release this perforin, they release this granzyme, and it killed this cell. Tumor necrosis factor, it kills cells. Interferon, we won't worry about everything that it does. But cytotoxic T cells, they're kind of like the bulldogs of your immune system. They don't mess around. They start killing cells. But what cell did they just kill? They killed our own body cells. So why is that good? Well, remember, this cell that it killed, that virus-infected cell, or maybe it was cancerous, but this virus-infected cell, there was no saving it. Remember, it was a lost cause. And if we let it go, it would produce more virus-infected cells. So killing this cell is to our body's benefit. And that's why that happened. But that still leaves those regulatory T cells. Now, regulatory T cells are kind of weird. And I'm going to present it in a way that isn't really true, but it's going to be a really simplified way of understanding it. So we saw the helper T cells. Helper T cells are telling your body, hey, something's here, we need to get into fight mode. And that activates our immune system, and our immune system goes around on the hunt, releasing lots of chemicals to kill things that don't belong there. But if that went on forever, eventually it would start harming our own body tissues. That would not be a good thing. We have to turn this off at some point. So helper T cells are on a timer. And as time passes, helper T cells change and become regulatory or suppressor T cells. Suppressor T cells then start releasing chemicals that say, stop fighting. Regulatory T cells turn your immune system back off. It takes it out of fight mode. And that's what we need so that we don't eventually start destroying our body tissues. Now, some types of autoimmune diseases result from regulatory T cells that are not active enough. So our immune system stays in fight mode and we actually start damaging some of our own tissues. Now that's not really the full story, but it's close enough to the full story that it gives you an idea of how regulatory T cells work. So this is from a different textbook, but it shows you the difference between Class 1 and Class 2 major histocompatibility complex. And here is a cancer cell being attacked by cytotoxic T cells. Now let's talk a little bit more about transplants. So like we said, transplants are given to people who need them. When we put an organ or a tissue into somebody, we're doing it because they need that to stay healthy. If someone gets a liver transplant, it's because their liver isn't functioning and they need that liver to stay alive. So the liver is helping them. But to your immune system, every time something is transplanted into your body, well, it is seen as foreign, and your immune system has no way of knowing that that organ or tissue is there to help you. All it knows is this organ or this tissue is out of place, it's foreign, and it will set off an immune response. And that's what happens when you hear about someone got a transplant and it was rejected. Their immune system attacked it, and it was no longer functional. So, if someone gets a transplant of any sort, they're going to have to be on immunosuppressants for the rest of their life. Immunosuppressants are a class of drugs. It's not a single drug. It's a class of drug that basically targets cells that go through a lot of mitosis, such as a lot of the leukocytes, like lymphocytes. So that will keep your immune system from attacking this transplant. But the problem with that is that means if you get sick, if you catch a cold or something, you have no defense against it. Because again, we're suppressing your immune system. So it's kind of a big deal if someone has an organ transplant, they cannot be around someone who is sick, which makes things like flu season potentially really dangerous. Okay, so now let's talk about humoral immunity. And humoral immunity is going to function similar but different from cell-mediated immunity. So let's draw how B cells work. So let's go ahead and draw B cell activity. And what I've drawn here, here's our big, exaggeratedly large bacteria again. And what we're drawing, again, could happen anywhere in the body. It's probably happening in your lymph nodes, but it could happen anywhere in your body. So, across the top, these are our naive B cells. Now, they only have the receptor. They don't have that extra arm. And that's because B cells are capable of recognizing free antigen. They do not need an MHC to present it to them. Remember, that's what that arm did on T cells, both types. So here are our naive B cells. And just like T cells, they're constantly bumping their receptors up against every surface in the body. So at some point, B cell here with the squiggly shaped receptor comes up, touches his receptor against it, it doesn't fit, so it leaves, still naive, and nothing happens. And that keeps happening. Eventually, round-shaped receptor here comes along, touches his receptor against it, and it fits. Now, we're not going to draw this next step, but I want to explain what happens. When this fits, that activates this B cell. It says, hey, I'm foreign. And the B cell responds by saying, okay, we need to do something about this. So the B cell actually rips this antigen off and internalizes it. And he has MHCs of his own. And he puts that antigen on the surface of his MHC, goes and finds a helper T cell. The helper T cell blasts him with interleukin-2 to fully put him into active mode. That starts the differentiation proliferation process. So we're not drawing that, but just know helper T cell is involved. Well, once this B cell is activated because he saw his foreign antigen, we're going to get proliferation, differentiation. We get memory B cells that go hide. Memory B cells go hide. They don't do anything at all right now. They're going to hang out in your lymph system until the same exact strain of bacteria shows up again. But the effector B cells are called plasma cells. we give them a very special name. The cells that are going to fight this fight, when we're talking about B cells, the effector cells, are called plasma cells. So what do plasma cells do? How do they fight the current fight? They start secreting antibodies. And the antibodies are shaped so that they fit against the antigen that this B cell is specific to. So look at the receptor of the B cell that fit this antigen. Look at the ends of the antibodies. Same shape. Because now these antibodies are going to go into circulation, and every time they find one of these antigens, they're going to stick to it. So that's what B cells do when they're activated. They produce antibodies against their specific foreign antigen. So with B cells, they recognize free antigen. That means that it's just in your body but outside of your cells. It must be outside of a cell, because if it was inside of a cell, the B cell would not be able to recognize it. Differentiation is activated by helper T cells. So even though we didn't draw it, remember, he takes a piece of that antigen, goes to a helper T cell, gets blasted with interleukin-2, then he starts to go through that division process. They make memory B cells that go hide, and they make effector B cells called plasma cells. We don't just call them effector B cells. We call them plasma cells. What do the plasma cells do? They secrete antibodies shaped to fit the antigen that that B cell was specific to. So here's how your book draws that process. This is on page 831 of your textbook. Here are our different types of naive B cells. Here are their receptors. Here's the antigen that binds to this receptor because it's shaped right. It doesn't bind to these receptors. It doesn't bind to these receptors because they're the wrong shape. So this B cell takes a piece of that antigen, goes and finds a helper T cell. Here we see that MHC that the B cell has. B cells are antigen-presenting cells, so they have MHC class 2. They also have MHC class 1. Remember, every cell has MHC class 1. But B cells have MHC class 2. They go and show this antigen to the helper T cell. Helper T cell releases interleukin-2, and now this B cell starts to divide. And then when he starts to divide, he makes plasma cells that fight now by releasing antibodies. Look at the shape of the antibodies, the same as what we had up here. And memory B cells that go and hide, but they're ready to fight. They will start to become active immediately if this same antigen shows up in the future. So here is a naive B cell, and here is a plasma cell. You can see they look very, very different. Plasma cells have a really extensive rough endoplasmic reticulum. Now, I didn't make this a thinking cap question, but let's go ahead and use it as one. Why would the plasma cell have much more of a rough endoplasmic reticulum than the naive B cell does? Well, what class of what class of macromolecule are antibodies? They're proteins. Where are proteins made? If they're going to be secreted from the cell, they're made on the rough endoplasmic reticulum. So it's very, very extensive in a plasma cell. So this is similar to something that's on page 833 of your textbook, but I don't like the version that's on page 833 of your textbook. This is a much better representation of what I want you to know. This is showing something called the antibody titer. Really all that means is how much of the antibody do you have circulating in your plasma? So what we see down here is the days since encounter with an antigen. And now it doesn't matter which antigen. Let's just say antigen A. Now that's not necessarily antigen A that's on erythrocytes. This is just an antigen when it first shows up in your body. And almost a week later, we start to develop some antibodies to whichever antigen that was. But it's over a week later, about a week and a half later, before we have any real rise in that antibody count. So it's looks like about 10 days since we initially encountered it. Well, during that 10 days, you feel pretty miserable. That's when you're sick. And then after the illness goes, that antibody count will start to drop back down. Now it shows here on day 28, but this could be, next year. This could be a few years from now. It doesn't matter. Some point in the future, if that exact same antigen-carrying pathogen shows up in your body again, look what happens. Right here is where that second encounter happens. Remember up here we had almost a week before we had anything, and then it was kind of a slow rise? Here, there is no delay, and look how quickly it rises, and look how much it rises. This is almost immediate, and it is a very big response. If this were to happen a third time, same thing. A fourth time, same thing. When something shows up the first time, you get sick, and it's a while before you get a response to it. During this time, this is when we see all of what we just drew with the B cells. But now those memory cells are ready. And if this exact same pathogen shows up again, those memory cells, they don't take a week and a half to really start pumping out antibodies. They go into fight mode immediately. They convert to plasma cells, and right here, as soon as that pathogen shows up, they start secreting antibodies. And they don't just secrete antibodies, they secrete a lot of antibodies very, very quickly. So here we have a really exaggerated response. You don't get sick that second time or any future time. You only get sick the first time it shows up. And look what else happens. Those antibodies, they do drop a little bit, but not much at all. Those antibodies stay elevated in your blood. So memory cells and antibodies keep you from getting sick a second time from the same pathogen. But it's only good for the same antigen-carrying pathogen. This purple line right here shows what happens if a different antigen-carrying pathogen shows up. So it says, to antigen B. I wish they used a different naming scheme because this makes it sound like we're talking antigen B that's on blood. This is just antigen 2. Let's call this antigen 1 and this is antigen 2. If a different antigen shows up, it's just like what we saw over here. It's almost a week and a half before we get any noticeable effect. And then it drops back off. But if antigen 2 showed up again over here somewhere, then we would get a response that looks like this. So the first time an antigen shows up, you're going to get sick and you're going to have a small response. But you make memory cells. If that same thing shows up again, memory cells and antibodies exist to keep us from getting sick a second time. But what do those antibodies actually do? All we've talked about with antibodies is that they stick to things. And we did see some agglutination when we were talking about blood, but really all we say is antibodies stick to things. Well, let's see what antibodies do. Well, antibodies do stick to things. And that's all that they do, is they stick to things. But when they stick to things, they cause other stuff to happen. So one thing is neutralization. Remember how the viruses, I said that they're very specific, they stick to receptors on your cell surface because they have receptors that fit? Well, if we coat those viral receptors with antibodies, the virus has no way of sticking to anything and it can't make you sick. Bacteria, those antigens that we drew on the bacteria, that's how they stick to different parts of your body, and that can ultimately lead to you getting sick. So if we coat things like bacteria or viruses and similar other pathogens, then we neutralize them. We make them so that they can't attach and they can't do anything harmful. We saw with blood typing that it can cause agglutination. This is showing you what happened with that agglutination. Causes things to stick together. If these were bacteria instead of red blood cells, well, you can imagine that these bacteria being clustered together like that, they can't function the way that they normally would, which could potentially lead to you getting sick. Precipitation. Some antigens are not necessarily a part of a bigger pathogen like a bacteria or a virus. Some antigens dissolve in your plasma. Well, antibodies can still stick to them. And when the antibodies stick to them, they make larger chains like we see here. And that causes them to become insoluble. They settle out. They precipitate out. And this could lead to something like phagocytosis, or it can allow your kidneys to remove it so that you can urinate it out. Over here we see that some antibodies can trigger the complement cascade. Remember that from a couple of lectures ago? Some antibodies can trigger the complement cascade. So these are what can happen when antibodies bind to an antigen. But none of these kill the pathogen. Every single one of these, all that's happening is the antibody is sticking to the antigen, and that's it. Antibodies don't kill, they just stick. But look what happens when the antibodies stick. It can lead to phagocytosis. Remember opsonization? It can lead to inflammation and all of the things that come after inflammation. Remember that? It can lead to lysis of the cell. So even though the antibodies don't kill, all they do is stick. They can lead to something that does kill the pathogen. So that brings us to a thinking cap question. Explain the following statement. You've had a cold more than once. You've probably had strep throat more than once. You've probably had the flu more than once. But you've never had the same cold twice. You've never had the same strep twice. You've never had the same flu twice. So explain that statement. I will say that statement is true. You've never had the same cold twice. You have never had the same flu twice. Even if you've had the flu twice, it's different. So how is that true? Well, that's because of everything that we just talked about. We have memory cells. We have antibodies. And those things keep us from getting sick a second time. If you've ever had the flu, well, you now have memory and antibodies against that strain of flu. If you get the flu again, it's a different strain, and that different strain has different antigens. It might feel the same, but it's not the same. It's a different strain of flu. So we saw this in the previous lecture, but I said we would come back to it, and that's because it really makes more sense to cover it now. And that is, how do we develop humoral immunity? there's different ways to get humoral immunity. There's 4 different ways, but they fit into 2 different classes. We can get it through an active process or a passive process. And the difference is who or what made the antibodies. In an active process of humoral immunity, you made your own antibodies. In passive, someone or something else made the antibodies and you're just benefiting from them. So let's see the 4 different types. The first is naturally acquired active immunity. Naturally means that this is something that does occur in nature without any help. Active, again, means you made the antibodies yourself. And the best example of this is you got sick. You encountered the antibody or the antigen and started making your own antibodies. Like we just said, if you catch the flu, you encountered that pathogen that has antigens on it, and you started making antibodies against it. That's natural, and that's active. You made the antibodies. Over here, artificially acquired active immunity. Artificial means that this is something that does not occur on its own in nature. But again, active, you are making the antibodies yourself. So a perfect example of this is a vaccine. The way vaccines work is you are injected, so there's the artificial part. Injections, while they are good, they are not natural. So in a vaccine, you are injected with either a dead pathogen or a weakened pathogen. It's called attenuated. That just means that the parts of it that make you sick have been removed. Or pieces of a pathogen. So we just take some of the antigens off of a pathogen and inject those into you. In any case, the parts of the pathogen that make you sick are either not there or not functioning. So they're not going to make you sick. And once these pieces are injected into you, that has the exact same effect as if it showed up alive. Your immune system sees the shape. Your immune system responds by making memory cells and antibodies. So you're making the antibodies yourself after having been artificially exposed to the pathogen. Now over here on the passive side, someone or something else made the antibodies that you are benefiting from. First, naturally acquired. So this is how mom helps baby. And there's 2 examples of this. When mom is pregnant and she's carrying the baby, her antibodies cross the placenta and they keep baby safe while baby is inside of her. And when baby is born, her antibodies are still circulating in baby. Baby's not making his own antibodies yet because he's not been exposed to anything yet. But baby is carrying mom's antibodies for a while until they degrade. And those antibodies keep baby safe. Now, that is a natural process. But it's passive because baby didn't make the antibodies. Baby is just benefiting from mom's antibodies that are inside of baby. Now, after baby is born, maybe mom chooses to breastfeed. And breast milk carries mom's antibodies. So she is passing more of her antibodies into baby through her breast milk. And those antibodies continue to protect baby even after he's been born. Again, natural, it's natural to breastfeed. Passive, because baby did not make those antibodies. Baby's just benefiting from them. And the final type is probably the trickiest type. And this is artificially acquired passive immunity. Artificially acquired because, again, this is injection. but we're not injecting the same thing that we saw over here. Here, we are actually injecting antibodies. And a good example of this is, let's say you go hiking in the woods and you get bit and you look down and you see that it was a black widow that bit you. So you go to the hospital and the hospital asks you what it was. Hopefully you saw what bit you because that's going to be important. It was a black widow. It was a brown recluse. It was this type of venomous snake. But if you know what it was that bit you, then they're going to inject you with something called anti-venom. So let's see what anti-venom is. Anti-venom is developed using either a rabbit or a horse. I don't know why that is, but that's what they always use, is either a rabbit or a horse. And they will take this rabbit and inject it with a little bit of a specific venom, maybe black widow. If it's a different, like maybe brown recluse, they use a different rabbit. If it's a certain type of venomous snake, they use a different rabbit. So every rabbit that they use will have a specific venom that they're injecting. But they only inject a tiny amount, not enough to kill the rabbit, just enough to expose the rabbit or horse. And this poison, well, it's got antigens on it. So the rabbit or horse respond by making antibodies against that antigen on the venom. And then later on, they inject a little bit more. And later on, they inject a little bit more. Always the same type of venom. So what happens is this rabbit or horse starts developing lots of antibodies against this venom. And they take the blood, they draw off the antibodies, and they store them. So they have antibodies against black widow venom. They have antibodies against brown recluse venom or against this type of snake. So that way, if you get bit by a black widow, you go and they say, what bit you? You say it was a black widow. Well, they're going to inject you with antibodies that will bind to that black widow venom and prevent it from hurting you anymore. Now, when you get bit by something venomous, you will start making antibodies against it. But you're not going to make them quick enough. And you are going to possibly die before you have any real level of antibodies. This is just bypassing that process. Now, another type of artificially acquired passive immunity is, let's say you have intercourse with someone and you find out later that they had a certain type of hepatitis. Well, you could potentially get that hepatitis unless you go to the doctor and you say, Doc, I had intercourse with someone, they have hepatitis, and the doctor will give you an injection. And what they're injecting you with is antibodies that maybe someone else who had hepatitis donated. And by injecting these antibodies against that type of hepatitis, it could potentially keep you from catching hepatitis. But in both of those cases, anti-venom or serum injections is what that other was called, they're injecting you with antibodies that somebody or something, like a rabbit or horse, made. And that's passive. So those are the 4 ways to develop humoral immunity. Changing gears just a little bit, still talking about humoral immunity, vaccines, yes or no? And I always like, when I'm doing this class in person, to kind of take a survey and have people raise their hand. Should you get vaccines? Should you not get vaccines? This is a topic that people feel very, very strongly about. So, even though I can't see you, let's go ahead and say, vaccine, yes or no? Cast your vote, and let's see. Well, what if I showed you this? And this is the cover of an article that was posted in a medical journal called The Lancet. And The Lancet is a very prestigious medical journal. And even though the word autism is not written in this, this is the paper that shows the link between autism and vaccines. This paper is what set that ball rolling, and it was published in The Lancet by a man named Dr. Andrew Wakefield. And after this paper was published, that's when people really started becoming kind of vaccine adverse. They're very hesitant to vaccinate their children, which is understandable if vaccines cause autism, right? Well, what if instead I told you that if you look this article up on the internet, you won't find this. What you'll find is this. Because after this was published, it was found that Dr. Andrew Wakefield faked the whole thing. And it is not Dr. Andrew Wakefield anymore. He had his degree revoked. He cannot practice medicine anymore. And he is just Andrew Wakefield, not doctor. He faked the entire thing. But now imagine you are a young mother, and you hear that vaccines cause autism, and it was published in a medical journal. Now, are you going to bother to find out whether that was real? Are you going to bother to find out whether that was redacted? Well, no, you're not. You're going to remember that, hey, medical journal said vaccines cause autism. And that set off the anti-vaccine movement, even though it was fake. So, let's jump on that and talk about vaccines and autism and all of the scary stuff that goes along with it. So, everyone has seen in the media that people like this genius shown here, Jenny McCarthy and other big-name celebrities, are very, very anti-vaccination. And over here on the left is a lot of their points. So, on one hand we have doctors and trained medical professionals, but on the other hand we have people with star power. So who wins? Well, let's see. Let's take these arguments in order. First, vaccines contain mercury, and mercury causes heavy metal poisoning. Yes, mercury absolutely causes heavy metal poisoning. But sodium is toxic, and it will kill you. Chlorine is toxic, and it will kill you. But everyone listening to me has had a lot of both sodium and chlorine in your food today. And you had it in something called table salt. Table salt is just sodium and chlorine. Just because something is toxic does not mean that it is always toxic in every form. Sodium chloride, table salt, is not toxic. I mean, it's not really the best thing to eat because of blood pressure, as we've seen, but it's not going to kill you if you eat it. So what does that have to do with mercury? Mercury is an ingredient in something called thimerosal. And thimerosal is an ingredient used in vaccines. But the thing is, the mercury that's used in thimerosal is a different type of mercury, and it is not going to cause heavy metal poisoning. As a matter of fact, there is more mercury in a single can of tuna fish than in all of the vaccines that someone will have in their entire life. But on top of that, thimerosal was removed from vaccines in the 90s. There's only one type of vaccine that has thimerosal in it, and it's hardly ever given. So when this thimerosal was removed, and there is no mercury in the vaccine, if this was the link, shouldn't autism have gone down? Well, it didn't. It actually went up. So let's move to the next point. Formaldehyde. Formaldehyde is what they embalm dead people with. And it is found in vaccines. So how can that be, good at all? Well, yeah, formaldehyde is used to embalm people. But formaldehyde is also a natural metabolite. you create formaldehyde when you eat food. When you eat an apple, your body makes more formaldehyde than is in all of the vaccines that you will ever take in your life. But formaldehyde is really quickly broken down by the body. In vaccines, it's just used as a preservative. But it is in such a small amount that it is, again, less than what you would have in an apple, and it is almost immediately converted into something completely harmless. So let's scratch that off and move to the next one. Antifreeze. Vaccines contain things that are in antifreeze, so that can't be good. Why would you put something in your body that you would put in your car? Well, no, it's not at all the same type of antifreeze. Again, this is a preservative. It just makes the vaccine last longer. But fish that live in cold water have antifreeze, and they're safe. The stuff that is used in vaccines is not the stuff that is used in car antifreeze, and it is harmless to you. So let's scratch that off. Timing. Well, I got my kid vaccinated, and just after that, he was diagnosed with autism, so it has to be the vaccines that caused it. There's a lot wrong with that statement. Possibly, your kid had autism way before this. Possibly, your kid doesn't really have autism. Or, even if your kid does have autism, just because your kid was diagnosed around the same time as these vaccines, correlation does not mean causation. The vaccines, just because they occurred around the same time, doesn't mean that they are the cause. See, vaccines, as we've just seen, do not contain anything that has been shown to cause autism. But this first round of vaccines, or the second round of vaccines, or the third round of vaccines, they occur at a specific schedule. But with autism, we are getting really, really good at diagnosing autism. And every few years, we get even better. And we can diagnose children with autism at a much younger age. And right now, we can diagnose children with autism at around the same time that they're getting these vaccinations. So, timing, that's just a coincidence. Maybe your kid does have autism, but that's just because we're really good at diagnosing it now. And it's just around the same time that some of these vaccines are given. Okay, so scratch that off and move to the next one. Vaccines don't cause autism. But even if they did, which they don't, let's just pretend that they do. But again, they don't. But for sake of argument, if vaccines caused autism, is that really such a bad thing? Because if you don't get vaccinated, a lot of these things that we're vaccinating a child against can kill the child or can lead to paralysis of the child. Is autism really worse than paralysis or death? No, it's not. But that point is moot because vaccines don't cause autism. So let's just take autism off the table and move on to the final point. I got vaccinated and I still got the flu. Or I got vaccinated and it gave me the flu. Neither of those things are true. So I got vaccinated and it gave me the flu? No. The flu, if you got the flu after getting a vaccine, one of two things happened. One, we're not 100% perfect with guessing which strain of flu is going to come around. And sometimes we vaccinate against one strain when a different strain comes through. And that strain is not going to be protected if you got a different vaccination. But what's the big deal there? All that means is now you're immune to something if it ever does come through. But at the same time, if you do get the vaccine against the correct strain of flu and you still get the flu, all that means is the flu was already there before you started developing your immune response. So, no, it did not give you the flu. You already had the flu. You just started showing symptoms after you got the shot. I got the vaccine and I still got the flu. Yeah, that's maybe true. Maybe you did. But again, maybe you were vaccinated and a different strain came through. Or maybe you were vaccinated and you didn't develop your immune response in time and you still got the flu. But did the vaccination harm you? No, it didn't. But the real thing about vaccines isn't so much what it does for you. It's what it does for everyone around you. The more people that get vaccinated, the better chance elderly people or infants or people who are immunocompromised and can't get the shot, the better they're protected. Vaccinated people create a barrier around these people who can't get vaccines. That's called herd immunity. And vaccines absolutely work. So what this shows us on the left is the rate of death from a certain disorder before the vaccine, and over here after the vaccine. This shows us diphtheria, 100% eradicated. Influenza, 99%. Hepatitis A, 91%. On and on and on and on. Polio, 100%. Smallpox, 100%. These are the rates of drop after the vaccine was introduced. So the answer to that question earlier, it's not just yes, it's hell yes. Absolutely get your vaccines. Especially if you're going to be working in a medical setting around people who might not be able to get vaccines. Don't go to work and bring the gift of death to an old woman who can't get the vaccine or whose immune response isn't strong enough to a vaccine. Don't get it so that you don't get sick. While that is a great point, vaccines will often keep you from getting sick. But vaccines might save that old lady or that old man or that little baby from getting dead. And in the end, this is the best statement that I could think of regarding vaccines, and that is vaccines, of all the stuff that they don't cause, what they do cause is adults. Okay, so now moving on to something a little bit lighter, back to just immune response in general. This is again from a different textbook. And now we've talked about the innate immune response, the first line, the second line of defense. We've talked about the adaptive immune response with cellular immunity, with humoral immunity. And we've talked about them one at a time. And it kind of gives you the idea of, well, if this doesn't work, then we move to this. And if this doesn't work, then we move to this. But this shows us if someone inhales something, then what does your body do? It does everything at the same time. It does not wait to see if something works. It throws the whole arsenal at it at once to make sure you don't get sick. Over here we see the mucus and the cilia and stuff like that that are part of the first line of defense. But we also see the second line of defense and several different ways that are thrown. Over here we see the third line, the adaptive defenses, and all of the different mechanisms used to prevent us from getting sick. All of this happens at once, rather than one at a time. And again, it all depends on helper T cells. Here's that helper T cell and all of the stuff that it releases and how those things activate different branches of your immune system. But it's the helper T cell, that little guy that didn't do any fighting of his own, the little guy that just rang the alarm bell. He's the one that is absolutely the most responsible for our immune response. Now let's change gears and talk about allergies. And there are different types of allergies. This is just kind of generalized. So let's see how allergies work. Now, there are different types of allergens, but they're all based on antigens. But typically, allergens are something that really do not harm our body. Previously, when we've talked about antigens, we've talked about something like on a virus or on a bacteria, something that could potentially do us damage. But now we're going to talk about something like pollen or peanuts or shellfish, something that really isn't harmful. But if you have allergies, this is kind of a miscommunication, and your immune system thinks that it is harmful. So I'm going to call this ragweed pollen. Now, it could be any number of allergens. I'm just using ragweed pollen as the example. So the very first time you are exposed to ragweed pollen, if you are allergic to it, this is what happens. The very first time you're exposed to it, you breathe that ragweed pollen into your body. And a B cell has a receptor that fits that ragweed pollen. And that tells your B cell, hey, this is something that could potentially do us harm. And the B cells, they go through that process that we drew out, and the plasma cells start secreting antibodies. Lots of them. And now we've seen the stuff that antibodies can do, but there's something else that they can do. They will attach to B cells, they will attach to mast cells. We talked about those back in Bio 137 in the skin. So these antibodies will attach to the surface of mast cells and to basophils. Now, basophils and mast cells, they are really, really similar. They do a lot of the same jobs. So those antibodies attach themselves to mast cells and basophils. Fast forward to the next year, and it's ragweed season again. Now, up to this point, nothing happened. You didn't develop symptoms or anything the first time that you breathed that allergen in. But now this is the second time you breathe in some ragweed pollen, or the 20th time, or the 500th time. You breathe that pollen in, and now it's going to attach to those antibodies that are on the surface of those mast cells and basophils. And here's how those cells respond. When a mast cell with antibodies on its surface bind whichever antigen, in this case, ragweed pollen, that's going to trigger the mast cell or basophil to start releasing huge amounts of what? What do you think of when you think of allergies? Histamine. Histamine is poured out into your tissues. Histamine causes things like vasodilation, and those capillaries become very permeable. That causes swelling, like in your sinuses or like behind your eyes. It causes release of mucus, which causes you to get all snotty, like up inside your nose or down in through your bronchi. It causes bronchoconstriction. Remember, bronchoconstriction, that is different from vasoconstriction. Bronchoconstriction. Those small airways down in your lungs, they get smaller because of that swelling. Makes it harder to breathe. All of the stuff that you associate with an allergic reaction. If you have hay fever, this is what's causing all of that. So what do? You take antihistamines. And antihistamines block the histamine, so all of this stops happening. But let's see what happens on a bigger scale. Let's say you have a peanut allergy, or let's say you have a shellfish allergy. And if you have a shellfish allergy and you eat some shrimp, well, this all still happens, but on a much bigger scale. You get dramatic vasodilation and permeability is increased, so your blood pressure drops a lot. Maybe you pass out. You get severe release of mucus. It starts to choke you. You get severe constriction of those bronchioles, and you can't breathe. This is anaphylactic shock. You pass out, you start to suffocate. It can kill you. So what do? They inject you with an EpiPen. Epinephrine and certain steroids undo all of this. So that's why if you have some sort of severe allergy, what do? You carry an EpiPen with you so that this doesn't all happen. And lastly, let's talk about autoimmune diseases. There are different types of autoimmune diseases. Autoimmune disease is a class. It's not a disease itself. So the thing that's common among autoimmune diseases is your body, your immune system, sees part of yourself as foreign. And there's different types of autoimmune disease, such as cross-reactivity. In cross-reactivity, we have an antigen in one area that looks similar to an antigen in another area. So, for example, there's something called rheumatic fever that ties in with strep throat. The bacteria that cause strep throat have antigens that are kind of shaped like antigens on parts of your aorta and parts of your bicuspid valve. And when you start to mount an immune response against strep throat strains, sometimes your immune system will start attacking your aortic valve and your bicuspid valve. And that's what rheumatic fever is. Abnormal exposure of self-antigens to blood. Well, one good example of this is in males, there's something called the blood-testis barrier. Males do not start making sperm until they hit puberty. When they are first born and up through, early adolescence, they don't make sperm. So when they start making sperm, any antigens that are on those sperm cells look brand new. Your immune system does not recognize them as self. They would recognize them as foreign, and they would attack them. So your testes actually have something similar to the blood-brain barrier, but it's called the blood-testis barrier. The sperm cells are in an area of the testes that are blocked off from the immune system. That's actually why a lot of testicular cancer can be very difficult, because your immune system doesn't have access to certain parts of the testis. But the blood-testis barrier prevents your immune system from seeing the sperm cells as foreign and attacking them. Change in structure of self-antigens. Sometimes antigens throughout someone's life can slightly change shape. But that leads to the problem of, if they change shape, now they look out of place. And if they look out of place, the immune system will attack them. Some evidence seems to say that maybe some types of type 1 diabetes mellitus is caused by this. Now, there are other causes, but some evidence seems to say that some types of type 1 diabetes mellitus occur because of this change of structure. And sometimes it's because of just a mishap during the maturation process. Maybe the cells fail to pass self-tolerance, but they are not eliminated by negative selection. They should be eliminated by negative selection, but they're not. and they go on to develop and mature and go into circulation. And then they might go off and attack some part of your body. So now let's talk about HIV and AIDS. HIV is the virus that causes AIDS, the disorder. And before we can talk about how HIV/AIDS works, we need to go back and talk a little bit more about viruses in general. And specifically, we're going to talk about something called retroviruses, which is what HIV belongs to. Now, let's go even further back and talk about something from Bio 137, which may or may not have been covered. It was covered, they just might not have used this phrase. There's something called the central dogma of biology. And all that means is the central idea that biology is founded on. And that is that all living things, from the lowest of bacteria through plants and fungi and insects and humans and everything in between, they all do the exact same thing. And that is, they start with DNA. From that, they make RNA. And then that RNA is read, and we make proteins, which give us our traits. So that is the central dogma of biology, DNA to RNA to protein traits. Now, retroviruses, they do a little something different. Retroviruses contain RNA. So we're going to call that viral RNA. And that's what we see. Over here is our virus, and here's the RNA for that virus. And they inject their RNA, as we saw a lecture or two ago. They inject their RNA into the cell. And that viral RNA is actually converted to viral DNA. Now, let's go up here and say all living things read DNA and they make RNA from it. That's called transcription. That reading DNA and making RNA from it is transcription. Well, down here, RNA is being read and converted to DNA. That is not something that living things normally do. So this is the reverse of transcription. This is reverse transcription. Now, humans and any living thing for that matter is not equipped to do this. But in the viral genes, it contains information to trick your cells into making an enzyme called reverse transcriptase. And the reverse transcriptase is what causes the viral RNA to be converted to viral DNA. And the viral DNA is placed into the host DNA. Remember when I said someone with a virus will forever have that virus? That's why. Especially when we're talking about retroviruses, that viral DNA becomes a part of the host cell's DNA. And now that viral DNA, the genes for that virus, will then be converted to RNA and converted into the proteins needed to make more of that virus. And that's how HIV works. HIV injects its viral RNA. And then that viral DNA is produced, and it becomes a part of the person's DNA. And the person's DNA now contains genes for that virus. More copies of that virus are made. Now, viruses that do this, since they're using a process that doesn't normally occur in the cell, it's using an enzyme that's not native to that cell to do something that does not occur in that cell. It's reading RNA and making DNA from it. Well, cells aren't equipped to do this, and they're not very good at it. So every time they do this, there are some errors. And that DNA that's produced has some incorrect spots in it. Remember, DNA is just a series of A's and T's and C's and G's in a certain order. And every time we make this DNA using the reverse transcription process, some wrong letters are laid down. So we end up with weird-looking strands of DNA. But that means if we change the DNA, we change the RNA that results from it. And if we change the RNA that results from it, the protein that results from that is going to be shaped a little bit different. Every time we change the DNA, the protein is going to be a little bit different. So every single time HIV goes through this process, the proteins are shaped a little bit different. Proteins such as these antigens on its surface. And if the antigens on its surface are shaped a little bit different every time a new copy of that virus is made, what does that mean? It means that the immune system doesn't know what to look for. If it runs into a copy of this virus, it starts looking for things that are shaped like these antigens. But then when the next batch of that virus is made, the antigens are going to be shaped a little bit different. So the immune system is constantly looking for a previous version of this virus. That's also why no vaccine can be made. The vaccine would put in pieces of these antigens, but those pieces of antigens show what the virus used to look like, not what it looks like now. Now that said, we are getting really close to making a vaccine against this. HIV is right about to have a vaccine for it. We're not there yet, but we're getting really close. Because even though these antigens are shaped different every time, there are certain areas of this virus that don't change quite so much when this process occurs. We say they are conserved. And if they don't change very much, then that allows us to have our immune system go looking for it. And we're almost there. But why is HIV such a problem? Remember when we said that viruses target very specific species? What affects a cat may not affect a person. And then we also said that some viruses are more specific than that. While they may affect heart cells, they might not affect bone cells. Well, HIV targets CD4 receptors. And where are CD4 receptors found? On helper T cells. So this virus attaches to CD4 receptors, and when it gets inside of a cell, it does what all viruses do. It hijacks that cell. It preoccupies that cell with making more copies of the virus. So CD4 cells, helper T cells, stop doing what they're supposed to do, and they just start making more and more and more copies of HIV. What happens if we don't have helper T cells? Well, remember what I kept saying earlier? Helper T cells, if they don't do their job, the immune system is turned off. So something like the common cold or bronchitis, that's not a big deal if you catch it. Well, if you don't have helper T cells, then the common cold or bronchitis can be fatal. So HIV and AIDS doesn't kill, but it allows things that are really harmless to most people to become fatal, and that's what kills. Opportunistic infections is what those are called. And that is what kills someone with HIV/AIDS. Now, back in the 80s, the 90s, when someone was told they have AIDS, that was a death sentence. It was, okay, I'm sorry, you're going to die soon. But that's not the case any longer. Now, with one or two exceptions, when someone gets AIDS, they're going to have AIDS forever. There have been a couple of people who seem to have recovered. But, out of millions of cases, 1 or 2, we discount those. When someone has AIDS, they will have AIDS for the rest of their life. But at the same time, when someone has AIDS, they used to be told, okay, you're going to die soon. Now someone who finds out that they have AIDS, they can live as long as someone without AIDS. It's no longer a death sentence. Now, they will have to take very large doses of lots of different medicines for the rest of their life. It's a real inconvenience. They have to be extremely careful. But it's not a death sentence. They will live as long as someone without AIDS. Now, soon, we will probably have a vaccine. We will probably have a treatment. We're not there yet, but I hope in a future version of this lecture I can completely change this part. And that's how the immune system works. So, that is the end of this unit. Up next is Lecture Exam 2. And Lecture Exam 2 is going to primarily focus on respiratory, lymphatic, and immune. But remember, all lecture exams are cumulative. So older material is still fair game for these exams. All right, let me know if you have any questions, and I will talk to you next time.